Good evening to everyone. Myself, Dr. Veena CN, Associate Professor in the Department of Physiology at Dr. Chandramma Dayananda Sagar Institute of Medical Education and Research, Dayanand Sagar University. We'll be talking about minute ventilation. I thank the Karnataka Pulmonologist Association for providing me an opportunity to discuss various physiological aspects related to minute ventilation. Today, I'll be talking about external respiration, minute ventilation, dead space air, alveolar ventilation, factors influencing minute ventilation, regulation of minute ventilation, and clinical significance of minute ventilation. So before discussing about the minute ventilation, let us discuss few points on respiration. Respiration encompasses two separate but related processes, namely the cellular respiration and external respiration. Cellular respiration refers to the intracellular metabolic processes carried out within the mitochondria, which uses oxygen and produce carbon dioxide, thereby deriving energy from the nutrient molecule. External respiration refers to the entire sequence of events in the exchange of oxygen and carbon dioxide between the external environment and the tissue cells. It encompasses four steps, namely the mechanical act of breathing or ventilation, which we will be discussing today, in which air is alternatively moved in and out of the lungs, exchanging air between the atmosphere and the air sacs. Diffusion, wherein oxygen and carbon dioxide are exchanged between the air in the alveolar sac and blood within the pulmonary capillaries. The third step would be the transport of gases between the lungs and the tissues via blood. And the next would be the exchange of gases between the tissue cells and the blood, which will be discussed in the subsequent sessions. So coming to the minute ventilation, breathing is a dynamic process involving movement of air in and out of the lungs. Minute ventilation is the volume of air inspired or expired per minute. It is also called as pulmonary ventilation. It involves two processes, namely the inspiration and expiration. Inspiration refers to inflow of air and occurs when the intrapulmonary pressure falls below the atmospheric pressure. Expiration refers to outflow of air or breathing out of air, which occurs when the intrapulmonary pressure exceeds the atmospheric pressure. These changes in the intrapulmonary pressure are related to changes in the intrapleural pressure, which is brought about by the changes in the size of the thoracic cavity caused by the contraction of respiratory muscles, namely the primary inspiratory muscles, which include diaphragm and the external intercostal muscles, which act during quiet inspiration or otherwise called as tidal respiration. Accessory inspiratory muscles, which include sternocleidomastoid, scalene muscles, ala nasi, and small muscles of the head and the neck, which act during forced inspiration. Expiratory muscles, which include internal intercostal muscles, rectus abdominis, internal and external obliques, and transverse abdominis, which act during forced expiration. So the tidal inspiration occurs when the diaphragm and the external intercostal muscles contract. Diaphragm and the external intercostal muscles contract and increase the diameter of the thoracic cavity in all the three dimensions: vertically, transverse, as well as antero-posterior diameter. About the contraction of the diaphragm constitutes for about 70 to 75% increase in the size of the thoracic cavity. When the diaphragm contracts, the dome shape becomes flattened and the level of the diaphragm is lowered, thereby increasing the vertical diameter. Contraction of the diaphragm also pulls in the ribs, leading to the increase in the transverse as well as antero-posterior diameter. Whereas tidal expiration is a passive process which involves elastic recoil of the lungs, relaxation of the diaphragm and the external intercostal muscles, and contraction of the abdominal muscles, which pulls the diaphragm upwards, thereby decreasing the size of the thoracic cavity. Minute ventilation can be measured by asking the subject to breathe through a valve box and collecting all the expired gas in a large bag. It can also be measured using Wright's respirometer. If other respiratory parameters like tidal volume is known, minute ventilation can be calculated using the formula VE equals VT into F, where VE is the minute ventilation, VT is the tidal volume, F is the frequency or the number of breaths per minute. For example, at rest, if a normal person moves approximately 500 ml of air, that is the tidal volume, in and out of the lungs with each breath and takes about 12 breaths per minute, the minute ventilation would be 6000 ml. Though the minute ventilation is 6000 ml, not all the air is available for the exchange due to the presence of dead space air. Dead space air is the portion of minute ventilation that does not take part in the exchange of gases. It is of three types: anatomical dead space, alveolar dead space, and physiological dead space. Anatomical dead space refers to the volume of the air which is present in the conducting zone, which extends from the nose to the terminal bronchioles. Alveolar dead space refers to the volume of the air present in those alveoli that does not take part in the gas exchange, whereas the physiological dead space includes both anatomical dead space and alveolar dead space. In normal healthy individuals, physiological dead space almost equals anatomical dead space. But however, in certain disease conditions with many non-functioning alveoli, the physiological dead space may markedly increase, sometimes as much as 10 times the anatomical dead space. Thus, the volume of the fresh air entering the alveoli during each inspiration equals the tidal volume minus the dead space air. From the previous example, if the tidal volume is 500 ml and the anatomical dead space is 150 ml, then the volume of the air entering the alveoli with each breath could be 350 ml, and the total volume of the fresh air entering the alveoli per minute would be 350 into 12, provided the respiratory rate is taken as 12 cycles per minute, would be 4200 ml, which is called as alveolar ventilation. Alveolar ventilation refers to the total volume of the fresh air entering the alveoli per minute. It is expressed as VA equals VE minus VD into F, where VA is the alveolar ventilation, VE is the minute ventilation, VD is the anatomical dead space, and F is the frequency or the number of breaths per minute. Thus, the alveolar ventilation rather than the minute ventilation is more important factor in determining the effectiveness of the gas exchange, which is evident from the following data. Here, the breathing pattern in subject A is rapid and shallow. Subject B breathes normally, and in subject C, the breathing pattern is slow and deep. Each subject has the exact same minute ventilation as it is evident in the slide, but that is, they move the same amount of air in and out of the lungs per minute, but when the dead space ventilation is subtracted from the minute ventilation, there is marked difference in the alveolar ventilation. In subject A, there is no alveolar ventilation, whereas in subject C, there is considerable greater alveolar ventilation. Several factors influence minute ventilation, which include partial pressure of carbon dioxide, partial pressure of oxygen, exercise, sleep, pregnancy, blood pressure, nociceptive stimuli, hypoglycemia, and temperature. So now let us discuss the effect of partial pressure of carbon dioxide on minute ventilation. The relationship between the partial pressure of carbon dioxide and minute ventilation is almost linear in the range of 45 to 80 mm of mercury. The minute ventilation increases by 2 to 5 liters per minute for every 1 mm increase in partial pressure of carbon dioxide. So increased partial pressure of carbon dioxide is sensed by the central chemoreceptors, leading to the increase in the tidal volume and the respiratory rate. Whereas the changes in the partial pressure of oxygen is sensed by the peripheral chemoreceptors, leading to the increase in the tidal volume and respiratory rate. So these receptors usually sense the oxygen tension rather than the oxygen content. Decreased partial pressure of oxygen increases the minute ventilation, but in case of partial pressure of carbon dioxide, increased partial pressure of carbon dioxide increases the minute ventilation. So coming to the exercise, minute ventilation increases with the level of exercise. It increases linearly with both oxygen consumption as well as carbon dioxide production up to the level of about 60% of the subject's breathing capacity. Above that level, minute ventilation increases faster than the oxygen consumption but continues to increase proportionately with the increase in the CO2 production. The increase is usually a result of increase in the tidal volume and the respiratory rate or the breathing frequency. Initially, the tidal volume increases more than the breathing frequency, but as the metabolic acidosis sets in, the increase in the breathing frequency predominates. Minute ventilation is also affected by the state of sleep. During slow wave sleep, breathing frequency and the inspiratory flow decreases, leading to the decreased minute ventilation. Pregnancy is also associated with variation in the minute ventilation. Progesterone acts directly on the central integrative control of the ventilation, leading to increase in the minute volume and decrease in partial pressure of carbon dioxide. The rate of increase is linear in fashion, starts early in the pregnancy and ends up around 40% above the baseline. Blood pressure seems to have a variable effect on the minute ventilation. Hypertension decreases the respiratory rate and hypotension increases the respiratory rate. With sufficient acute hypertensive event, respiration can be briefly ceased. It was Bodger et al. In 1996 who said or demonstrated the effect of nociceptive stimuli on the minute ventilation. He showed that the nociceptive stimuli due to various stimulations lead to variable minute ventilation. It was shown that the somatic pain causes hyperapnea and visceral pain causes apnea. Hypoglycemia also seemed to have an effect on minute ventilation. In vitro studies suggest that the insulin-induced hypoglycemia stimulates the carotid bodies to increase the ventilation in proportion to the elevation in the metabolic rate. Temperature also affects minute ventilation by increasing the sensitivity of the peripheral chemoreceptors to the partial pressure of oxygen and increasing the sensitivity of the central chemoreceptors to the partial pressure of to pH or the hydrogen ion concentration. Okay, so having been discussed about the factors affecting the minute ventilation, now let us discuss the regulation of minute ventilation. The respiratory control system reacts very effectively to the alteration in the internal environment of the body. The inputs to the respiratory center could be chemical, mechanical, or inputs from the higher cortical centers. Partial pressure of carbon dioxide is probably the most important input to the ventilatory control system in establishing the breath-to-breath levels of tidal volume and breathing frequency. These inputs lead to the stimulation of special neuronal units called as chemoreceptors, which alter their activity, thereby providing information to the medullary respiratory center or the pacemaker, which may be either stimulated or inhibited based on the input. So these respiratory centers send signals to the respiratory muscles, thereby altering the activity of the respiratory muscles, leading to variable minute ventilation. So if we could quote an example of effect of partial pressure of carbon dioxide on regulation of minute ventilation. Increased partial pressure of carbon dioxide leads to increased firing of central chemoreceptor, which causes stimulation of medullary respiratory center, leading to increased firing of the neurons to the diaphragm and the inspiratory intercostals, thereby causing contraction of diaphragm and inspiratory intercostals, with the end result being increased minute ventilation. The spontaneous rhythmicity generated in the medullary respiratory center can be completely overwhelmed by the influences from the higher brain centers. In fact, the greatest minute ventilation can be obtained from healthy, conscious human subjects, which can be attained voluntarily, exceeding those obtained with the stimuli of severe exercise, hypoxemia, and hypercapnia. Altered respiratory rate and pattern often accompany a variety of disease states. These diseases frequently lead to alteration in one of the three kinds of feedback to the central respiratory cortical center. For example, pathological conditions altering partial pressure of oxygen, partial pressure of carbon dioxide, and hydrogen ion concentration alter the input from both carotid body and the medullary chemoreceptor, which result in the change in tidal volume followed by change in the respiratory rate. Therefore, lung diseases that cause acute hypoxemia to a level of less than 55 mm of mercury usually produces increased ventilation. In metabolic acidosis, increased hydrogen ion concentration in the blood leads to the stimulation of peripheral chemoreceptors, thereby increasing ventilation. However, due to restricted movement of hydrogen ions into the CSF, it fails to stimulate the central chemoreceptors. But the hyperventilation induced by the stimulation of peripheral chemoreceptors usually leads to increased CSF pH, thereby restraining minute ventilation. Interstitial diseases of the lung enhances the stretch receptor response, leading to rapid and shallow breathing. Congestive cardiac failure also produces similar effect. This could be partly due to decreased lung compliance, but also may be a result of stimulation of J receptors, which is right next to the capillaries. Influences from the higher cortical centers are known to be modulated with the diseases. Anxiety can increase the respiratory rate as well as the pattern. An acute hyperventilation syndrome derived from the higher center can maintain high minute ventilation in face of elevated pH. Understanding the respiratory rate and pattern is very important addition in dealing with many respiratory, cardiovascular, and neurological diseases. Understanding the role of rate and the tidal volume is also essential in managing individuals requiring mechanical ventilation. To summarize, minute ventilation is the volume of the air inspired or expired per minute, can be measured using Wright's respirometer or from other respiratory parameters. In fact, alveolar ventilation rather than the minute ventilation is more important factor in determining the effectiveness of gaseous exchange, since the portion of minute ventilation does not take part in gas exchange. Several factors affect minute ventilation, which include changes in the partial pressure of oxygen, changes in the partial pressure of carbon dioxide, change in the temperature and hydrogen ion concentration. Level of exercise, sleep state, blood pressure, and blood glucose can also alter minute ventilation. The respiratory control system reacts very effectively to the changes in the internal environment, thereby altering minute ventilation. Perhaps Partial pressure of carbon dioxide is the most important input to the ventilatory control system. But however, higher minute ventilation can be obtained from influences by higher center. Minute ventilation can be altered in a variety of clinical conditions, which include lung diseases causing hypoxemia in metabolic acidosis, interstitial diseases of the lung, anxiety, congestive cardiac failure, and acute hyperventilation syndrome. These are my references. Thank you. Have a pleasant night.